Okay, thank you so much for giving me the opportunity to talk here at the UCSF Open Mind Consortium workshop, Lightning Talk. My name is Julian Neumann. I'm an assistant professor for interventional and cognitive neuromodulation at Charité Berlin, Germany. I'm going to talk about the Medtronic device. I want to make sure in advance that you know that I have no conflict of interest and no affiliation with Medtronic of any kind. Now, since we are going to talk about deep brain stimulation, I'm going to want to do a quick introduction here. Most of you probably know what deep brain stimulation is. It is a very effective treatment for several movement disorders, such as Parkinson's disease, dystonia, or essential tremor. Currently, deep brain stimulation works by connecting an IPG that is implanted with cables going into the depths of the brain, and a chronic stable 130 hertz or slightly different frequency of stimulation is set and applied with a constant voltage or current at a few milliamps. DBS is arguably the most reproducible and robust neurotechnological treatment, and therefore I think it is a really unique platform for trailblazing next generation clinical brain-computer interfaces. In Parkinson's disease, we have been working on oscillatory features and oscillations that reflect symptom severity since a very long time, and we could identify a biomarker, especially for akinetic and rigidity symptoms in these patients, that is beta oscillations. So synchronization in the beta frequency band, typically 13 to 30 hertz, has been shown to be affected by medication and to reflect the current state of the patient. Now, with the first sensing-enabled chronic implantable device, we were able to track these beta oscillations over a period of up to 8 months. And we could show that these oscillations are stable and associated with the off medication state when the patient is in his worst symptom state, and is also even after 8 months suppressed by medication, and therefore reflects a good biomarker of symptom severity that can be recorded in real time. Now, this first, the whole idea of beta activity being reflective of Parkinson's disease symptoms, and next, the idea that these symptoms or these biomarkers can be used to adapt the stimulation to the concurrent symptoms has been pioneered by Peter Brown. And he has published a landmark study with an externalized experimental setup for bidirectional stimulation control. And this is the study published as first author by Simon Little. And it demonstrates how sandwich montage recording from depth electrodes and stimulating the contact in between can be used to adapt the stimulation to the increase of beta activity. So whenever beta crosses an upper threshold, the stimulation is turned on, and then beta goes down and the stimulation is turned off. This was shown to be effective as chronic stimulation, with only 50% of the current use, and significantly better than random pattern stimulation. Now, these first advances have really led to the development of a chronically implantable device that has come out this year, and we were the first center in the world that has implanted one of these devices. It's called Medtronic Percept. It's CE approved, FDA is still pending, and it's a theoretically bidirectional DBS device. I'm saying theoretically here because neither in Europe nor in the US the bidirectional control is currently allowed. That's because the approval requires a clinical study, and that is going to be initiated once FDA clearance has been passed. Now, what is exciting is that this device is fully implantable, can record brain activity over days, and in the future, this will allow us to use the oscillatory amplitude of biomarkers to adapt the stimulation in a large cohort of patients in multi-center trials. We have to admit here, and I hope that this was not a profit-based decision by Medtronic that this PERS device is a primary cell. That means that it's not rechargeable, and sensing and particularly streaming brain activity to a tablet costs battery and therefore costs days of therapy for the patient before a new replacement needs to be performed. Now, the technical specifications are quite similar to what we are used to from standard electrophysiology processes. It has a sampling rate of 250 Hertz, 16 channels, does an FFT with a window of 64 or 256 samples. You can record up to 6 channels simultaneously. Patients can trigger events and snapshots. There's a recording of stimulation capability using the little described montage from the Simon Little paper. And what is really exciting for us now is that it's the first time that we can do really chronic recordings over up to 60 days. And this is what the activity looks like projected using aDBS to the target. You can see the electrode and the stimulation VTA, and you can see the band power plotted concurrent with the current date of the recording and time. What you can see here from the first plots is that there's a huge variability over the day of these biomarkers, and these are things that we need to explore in the future to really make a bidirectional device worthwhile for the patients. Now, the recording experience was, as I said, pretty straightforward. The raw data looks really good, much better than I have seen from previous generations of sensing-enabled implantable devices. Also, the resulting power spectra demonstrate intricate features in low and higher frequency bands. But there are also artifacts, especially during stimulation. You can see cardioballistic artifacts, and I'm not sure how patients affected by this artifact can be included in a study that will look at adaptive stimulation. As far as I know, such patients would be excluded. That means that whenever you have a patient that has a cardioballistic artifact during stimulation, he could not receive bidirectional stimulation control, which is a huge issue, I think. Furthermore, movement artifacts can be a problem. Now, this is a recording from a very severely affected dystonia patient who has generalized dystonia and trunk movements of the cables led to these movement artifacts, which could also be very challenging to get hold of for adaptive stimulation in the future. So from a clinician's perspective, the conventional programming is really unaltered, but the recording systems are complex. I think we will really need neurophysiologists in the clinic to support clinicians in doing studies and also doing routine captures of brain activity. And this brings a new team together and in an interdisciplinary approach, which in some centers such as Berlin or UCSF is already established, but in many centers it is not. Now, for the patient, in addition to their normal experience, they can do snapshot recordings, which can be defined. Snapshots can have a name, and patients can be asked to do snapshots, for example, whenever they take the medication, when they feel they're particularly bad, or when they have peak dose dyskinesia. There are 2 adaptive stimulation modes, which will be able to be used in the future. A single threshold mode, as described by Little et al. In 2013, and a dual threshold mode, which has been used by other centers, for example, Alberto Priori, but also Helen Bronte-Stewart. And the dual threshold mode has an upper and a lower threshold. Whenever the beta activity is in between thresholds, stimulation is stable. But when a threshold is crossed, stimulation moves up or down independence of the signal. The theoretical bidirectional workflow is to record a full montage, look at the spectra and identify peaks and frequency bands of interest. Then you need to define the recording and stimulation channels and choose the adaptive control mode. And then you can adjust the stimulation settings and to look at aDBS, move the stimulation parameters. And this allows also recording of raw data during stimulation and aDBS in parallel. Now, having introduced the Percept and this single biomarker approach, we have previously characterized many different biomarkers and different symptom-specific aspects of oscillatory synchronization. You can read about this in our review in 2019, Neurotherapeutics. And then additionally, there have been other sensing modalities being introduced, especially at UCSF. Phil Starr has pioneered the use of electrocorticography for DBS research. Now, he has, using previous generation of Medtronic device, already shown that it is feasible to record cortical activity, in this case, gamma band activity that occurs during dyskinesia, to adapt stimulation parameters. Our center has a collaboration with Mark Richardson, Director of Functional Neurosurgery at MGH, Harvard University, to explore multi-feature approaches for adaptive DBS. And here we show our first results regarding movement decoding from cortex across patients. So we trained machine learning models, a variety of them actually, and looked whether it's possible to decode behavior across patients. And this is the decoding performance of grip force from a completely newly recorded patient in Berlin. So different center, different recording setup. We were able to very reliably and accurately decode the force that the patient was putting on the grip force lever based on a model that is trained on from other patients. So in the future, I believe that the most promising approach to aDBS will be multi-feature decoding based on machine learning models to identify states of the patient, dysarthria, freezing, walking, dyskinesia, and that should inform an adaptive control algorithm that identifies optimal stimulation parameters, as for example, pulse width, frequency, amplitude, or the active contact, to really make an impact in patients' experience with aDBS. And I want to say that all the source code is available openly on github.com neuromodulation that I am using, and also check out aDBS, which is the electrode localization software used in this talk. And I want to briefly summarize that Medtronic Percept is a bidirectional device. Feedback-controlled stimulation is not yet approved. It is a primary cell, so the battery will drain when you record, and I hope that there will be a rechargeable device soon. Single biomarker studies will be the first step and very important steps towards aDBS in movement disorders. The sensing parameters can be difficult and manifold, and we will have to establish them in multi-center trials. And ultimately, I believe that multi-feature decoding from cortical sensing may be a more promising strategy to impact therapeutic outcome for adaptive DBS. Thank you all. I have to thank also my collaborators, Andrea Kuhn, the Director of Movement Disorders and Neuromodulation Unit in Berlin, Timon, who has done the machine learning work in my lab, Mark and Phil, who are great collaborators, Andreas Horn and Coralie de Hinten, who are inspiring peers in my field. And I hope you enjoyed the talk and you have a great conference. Goodbye.